Hello, everyone. Welcome to my QuPath video tutorials. In this video, I'm going to show you how to perform multiplexed analysis using the machine learning method. You can see I loaded my demonstration data. For machine learning, we need to select an area that can represent the whole tissue. If we use the rectangle tool, we can select this area. I had a look at the tissue before. This area contains the positive staining for all the markers. We can have a look, use the bright contrast tool. You can see here. We can switch off all the channels first. Then we can have a look at each staining in this area. First is the CD31. You can see the area we choose have positive CD31 staining. Then we can have a look at the second staining, CD4. We can see CD4 positive staining. Next is CD44. Then pan-cytokeratin. You can see we also have the pan-cytokeratin positive staining. Next will be pCNA. You can see pCNA positive staining in this area. And also CD45RO. Next one is SMA. We also have positive staining in this area. We can go through other markers. You can see positive staining for HLA. Then Ki67. We maintain CD11 and CD8. Then CD20 collagen. You can see collagen positive staining in this area. The last one is protoplanin. We can also see protoplanin staining in the area we choose. So this area is good for us to train the images. So the reason I show you one marker by one marker is that you need to have a look at your tissue and select an area that can see all the positive stainings. So now we choose the annotation area. So first we need to perform the cell segmentation for the chosen area. I'm going to use the start-dist method. If you want to know the details, you can watch my previous video. Because in this one you can see the image, the pixel is 0.5, so we can use the 0.5 micrometers script. Now we can press run. And we need to select the start-dist cell segmentation model. You can see the files is here. We just open it. Now QuPath is running the cell segmentation analysis. The area we choose takes 3 to 4 minutes. So QuPath finished the cell segmentation. You can see it took 4 minutes and 26 seconds. So now we can close it. You can see cell segmentation in our selected area. So now we can save the data. So to create a classifier for each channel, it is difficult to do it on the same image. So QuPath has a function to duplicate the images. If we go to classify train image, here you can see we can create duplicate channel training images. Let's do it. Now you can see you can select the channels that you want to duplicate the image. Because I'm using my own laptop, QuPath recommends you only do 5 to 10 channels each time. Unless you are using high-performance computer, you can duplicate all the channels at the same time. For today's demonstration, I'm going to use CD31 pan-cytokeratin PCN Ki67 CD11C. We can switch off other channels. Of course, we need to keep the DAPI channel. So now we can create duplicated images for each channel. If we go to project, you can see here is the original image. We duplicated 6 channels: DAPI, CD31, pan-cytokeratin, PCNA, Ki67, and CD11C. Let's save the data again. Now we can start to create a classifier for each channel. Of course, we don't need to do the DAPI channel. The first one will be CD31. We can click the CD31 image. Okay, you can see here on the top, the image changed to CD31. And also we have the cell segmentation for the selected area on the CD31 image. Because we only do CD31 here, we can switch off other channels. We can just keep CD31 on. We can make it bigger. Let's switch off the segmentation. Now you can see the CD31 staining. We can make it even bigger. We also switch on the DAPI channel. Now we need our pointer tool. We can click the pointer tool. You can see now we have the pointer tool. We can press add. First we can click one CD31 positive and single cells. Now if you go to annotation, you can see we have one here. We can set this one as CD31 positive cells. What you need to do is select CD31, then set selected channels. Now you can see our annotation named as CD31. At the moment, we only have one cell selected. So QuPath recommends you do 15 to 30 point. So we can click CD31 positive cells. Those ones are very bright. We can click. And also we can select the weaker stainings. Now we can move to a different area. Make it even bigger. We can use the pointer tool to select more cells. Let's move to a different area again. We can select the weaker stainings. So now you can see we have 27 points. That should be enough. So we set the CD31 positive ones. We also need to set the negative ones. So we press add. You can see we have a new annotation. Now we can just click CD31 negative cells. You can see we have 1 point here. And we need to set this one as the negative one. We just set it as ignore. The old version will be other. So let's set selected. You can see now our annotation changed to ignore. Those cells will be CD31 negative cells. So we can use the pointer tool to select more cells. We can move to another region, select more cells. We can move again. So we did 28. That should be enough. Now we can switch off the pointer tool and we can go to classify object classification, then change object classifier. So now you can see for the object filter, we can select cells. For the classifier, we can use artificial neural network. For features, we are not going to measure all because here we are doing the CD31. So we can use selected measurement. Now you can select. You can see we have all the available measurement here. We can find the CD31. You can see that's all the CD31 measurement. We can select all. Then you press apply. But you don't have to select all because the CD31 is a membrane protein. You can just select a membrane. Or if you go down, you can also select cells. So for the demonstration, we can just select all. So next will be the all classes. We also use selected classes. If you press the select, you can see here we have CD31 and ignore. We just press apply. So for the training, because we are using the point tools, we are not using the area, so we can choose the point only. Now we can use the live update. You can see CD1 positive cells in yellow color. Now we can switch on the cell segmentation. If we press the button here, you can see we switch on the cell segmentation. So let's just use the cell boundaries. Now we can zoom out. You can see all the positive cells are in yellow color in this selected area. So we did a classifier for the CD31. Now we need to save the classifier as CD31. We can save. You can see we save the classifier as CD31. So we finish the CD31. Now we can move to the second one, pan-cytokeratin. We can double click this image. So it asks us save the CD31. We can press yes. Now you can see the image changed to pan-cytokeratin. Again, we need to go to the channels. Now we can switch off CD31, switch on pan-cytokeratin. Let's switch off the cell segmentation. You can see in our selected area, most of our cells are pan-cytokeratin positive. We can zoom in. So we need the pointer tool again. We can add, then press the pan-cytokeratin positive cells. Then go to the annotation. Now we can set this one as pan-cytokeratin positive cells. Set selected. You can see here our annotation changed to pan-cytokeratin. So we can continue to click. Let's move to a different area. We can move to the top. Let's move again. You should select some positive one with bright staining, and also you should choose some with weak staining. Now should be enough for the pan-cytokeratin. We need to press Add again to select the negative cells. Now we can select one and set this one as ignore. Then we continue to select around 30 cells. We can move to a different area. Let's move again. Okay, we have 26. That should be enough. Now we can switch off the point tool and go to classify object classification, train object classifier. Now we use cells. Then classifier will be artificial network. Then we do selected measurement. We just select pan-cytokeratin. So we can select all of them, then apply. For classes, we use selected classes. If we have a look, you can see just ignore and pan-cytokeratin. We click apply. Now we do point only. We can do live update. Now we can switch on the cell segmentation. If we zoom out, you can see those pan-cytokeratin positive cells. So now we can save this one as the pan-cytokeratin. Let's save it. So we did the second one. Next we can go to the third one, pCNA. We double click. It will ask us to save the pan-cytokeratin. We press S. So you can see the image changed to pCNA. We can switch on pCNA channel. Let's do a auto to see the staining. We can switch off the cell segmentation and zoom in. You can see the pCNA positive cells. Again, we need our point tool. We can add, select. Now we can go to the annotation, set this one as pCNA. Set selected. We can do more cells. Let's move to a different area. So we have 25 cells. That should be enough. Now we can add, select negative ones. And then we need to set this one as ignore. And then we can set selected. We can select more negative cells. We can move to another area. Click more cells. Those cells are negative ones. Let's move to another area. So we have 26. That should be enough. Now we can go to classify objective classification, train object classifier. We select cells, neural network, select the measurement. We need to select PCNA. PCNA. You can see we can select all, then apply. Also, it's points only. Now we can do live update. We can switch on the cell segmentation. You can see PCNA positive cells in our selected area. Now we need to save this one as PCNA classifier. Let's save it. Okay, we did 3. You can check if you go to classify object classification, node object classifier. You can see now we have the CD31 pan-cytokeratin PCNA. Because we select 5 channels here, now we can do the Ki67. So let's switch on Ki67 expression. Now we can zoom in. So you can see here still PCNA image. We need to double click it. So now it changed to Ki67 image. Now we can use the point tool, add, then we can click, then we set the annotation as the Ki67. We can select more. So we have 28. That should be enough. Next one we add and select the negative ones. Now we set the class as ignore set selected. We can continue to click to get more negative cells. So we have 28 as well. That should be enough. Now we can go to classify object classification chain object classifier. We select the cells, neural network, select the measurement. We select the Ki67. You can see that's all the Ki67. We select the all and apply. And we do points only. Now we can do live update. You can see all the Ki67 positive cells. The cell segmentation kind of changed to green color. So we save this one as Ki67. So let's save our data. We can close it. Now we can do the last one, the CD11. We need to double click it. Task save Ki67 again. Okay, you can see the image changed to CD11. Now we switch on CD11. Okay, you can see here we can use our point tool, add, select CD11 positive ones. It is more difficult for CD11. So we can set this one as CD11. Then we can click to get more positive cells. Let's move to a different area. So we have 29 cells we can add. To select the negative ones, we can set this one as ignore. Then we select more cells. If we also did 29 cells, now we can close the pointer tool and go classify object classification, chain object classifier. We select cells, neural network, selected measurement. We can just select CD11. We can select all, apply. Class will be selected class. You can see that's the CD11C and ignore. Now we do live update. You can see all the CD11 positive cells. The cell segmentation changed to the yellow color. If you move your mouse, you can see here the yellow color cells will be CD11 cells. If you move to a red cells, you can see it is unclassified. Now we can save it as CD11C. C. Okay, so we did all the channels. For 5 channels, we selected here. Now we can save our data. If we go to classify object, classification, node object, classify, you can see we have 5 classifier we did just now. We can select all of them. Then apply classifier sequentially. Let's run. Okay, it is done. What we can do now, we can fill the cells. Then you can see, we can see the yellow ones are CD31 and CD11 positive cells. We can see Ki67 cells are in green color. Pan-cytokeratin cells are in purple color. And also PCNA in dark red color. The rest of the cells are in red color. So you can see we trained the images, but at the moment, because we only did the cell segmentation for the selected area, if we apply our classifier, it only shows the cells in the selected area. If you want to do the whole tissue, what you can do now, let's save our data. Then we can go to project and select the original image. So now you can see from here, we are on the original image. What we can do now is we can do another annotation. use the polygon tool to select the whole area. Now we can run cell segmentation on the whole area. I'm going to use the statist method again. We can drop the 0.5 scripts, then press run, and select the model file. Open. So it is running. I showed you before. The whole tissue takes more than half an hour to perform the cell segmentation analysis. We can wait until it finished. We can come back to do the rest demonstration to see how we can use classifier to classify the cells on the whole tissue. Okay, we finished the cell segmentation for the whole tissue. You can see here for the whole tissue we have more than 733,000 cells. But for our selected area to train the classifier, we only have 95,192 cells. So now we can apply the classifier on the whole tissue. We can go to classify, object classification, node object classifier. Then we can select all of them, CD31, pan-cytokeratin, pCNA, Ki67, and CD11C. Now we can apply the classifiers sequentially. Because we are doing the whole tissue, it will take longer to run. Okay, it is done. You can see now on the whole tissue, we can see CD31 and CD11C cells in yellow color, Ki67 positive cells in green color, and cytokeratin positive cells in purple color. Then pCNA positive cells in dark red color. So you can see we only trained the classifier in this area, but we can apply our trained classifier on the whole image, which works really well. So for the demonstration today, I only did 5 channels. When you actually analyze your own image, you need to go through all the channels, but the process will be the same. Okay, that's today's demonstration. I showed you how to use machine learning method to perform multiplexed analysis. I hope this video tutorial can help your data analysis. Thank you for watching my video tutorials.